Good morning and good afternoon, everyone. My name is François Gianelli. I'm a director at VCLS, and today I am pleased to present the challenges of potency assay development for cell and gene therapy. This audio file is accompanying poster 288 as presented for the ISCT Paris 2020. It is often said that developing an appropriate potency assay is one of the major challenges of cell and gene therapy development, and not only for the CMC aspect, but also beyond, as a potency assay draws a link between the clinical part and the CMC part of your development. When we look back a little bit, potency assays are in fact not something that is new and related solely to biological products. Centuries ago, we used to talk about biological activities for every kind of drugs that could be tested on animals upon receipt. The reason for the switch toward other type of measurement, which are the strength or even the content measurement for small molecular entities, is because the analytical tools allow us to predict the expected effect based on content only. And this is something for which we are not ready yet for all types of biologicals, and in particular for cell and gene therapy, which, as we will see, present very specific challenges. For cell and gene therapies, the key element is we need to ensure that we will deliver to patients a product that is appropriately potent in order to justify the risk-benefit balance that is very specific to those products. And we need to be able to do this in a context where, first, the general understanding of the mechanism of action is usually incomplete, and the analytical tools at our disposal usually doesn't allow us to address all structural components of the product and to correlate that structure to the expected effect. Finally, we need to all understand that the potency assay is not only a regulatory requirement, but also the only way you have to define the dose for your product to be administered, as well as understand and capture part of your manufacturing that may impact the final product functionality. The next question is usually, where do we start? And as very often for cell and gene therapy products, we need to take an integrated approach because the developments are shorter and shorter, especially if we consider the accelerated development pathways or incentives that have been deployed by every regulatory authority for those products to be available for as many patients as possible, as fast as possible. So we have the PRIME, the breakthrough designation, the RMAT designation, etc. And those development strategies are now so short that there is only so much time for a developer to gather sufficient data to develop fully correlated assays. That's where the integrated approach comes into play. It is really, really important to think ahead of the design of the research proof of concept or the design of the initial non-clinical studies, and how we can already capture functionality of the products at this stage, start exploring the mechanism of action, identifying potential potency assay candidates at this early stage. Then the next step is usually to start building a target product profile, where potency assay will obviously be one of your main critical quality attributes. That part is important because now that we have an initial understanding, even if it is incomplete, of the product functionality or mechanism of action, we need to focus on the expected clinical effect. What is the purpose of that product once in patient? And from there, we need to try to understand what can be measured as part of manufacturing and release to make sure that the product we obtain at the end of the process has the potential to fulfill that expected activity. The final benchmark we can do to try to identify the appropriate potency assay is to check for the current state of the art. This is something that was very difficult to do 10 years ago because there were only so many products for ATMPs or cell and gene therapies that were under development or even on the market. So information wasn't available back then except for some very scientific literature. From then, first thing first, agencies published a number of guidelines that can help you to try to assess that potency assay requirements. At the same time, for the products that have been authorized over the last 10 years, availability of the assessment report can give you at least partial information of strategies that have been successful so far. More and more companies are no longer trying to develop their first product, so it's usually very good if you can salvage past developments and gather the expertise and the knowledge you've been managed to generate on an initial development to further justify the approach for your potency assay development. Finally, even if the three categories of ATMPs are cell-based product, tissue-engineered product, and gene therapy product, we also see emerging families of product. And the CAR-T family is a good example. Not all companies is using the same type of gene modification or the same process to obtain those products, but the general mechanism of action, if we set the target aside, is usually the same. So we will see emerge probably some more standard approach for potency assay for those products based on the number of developments that are on the way. And that's something that is likely to expand to other type of products. There have been multiple chondrocyte-based tissues to date also. This being said, when we look at what the guidance says and possibly what the literature allows us to understand, we need to have an idea of what makes a good potency assay. And also that's where we usually realize that we will not need one, but a matrix of assays. So on the next figure, what you see is what is usually recognized as a good potency assay performance characteristic. And it is acknowledged that one assay is very unlikely to give you all those characteristics. That's why a matrix approach is usually important, using orthogonal methods that will partially overlap and will offer you the possibility to be as close as possible to the mechanism of action. And that's usually where you see most of the qualitative methods, or more robust as a test method and analytical tool where you will see emerge good quantitative method, even if they are only surrogates of the clinical activity. So a good potency assay is expected to be representative of the mechanism of action, and that element is key because that's how you build the entire strategy, whether it's for characterization or for the set of specification you will draw from that characterization exercise. For the cell-containing products, it is important to address the cell populations that are contributive to the mechanism of action, and then we can discuss whether it is direct contribution, which are the cells of interest, or if it's a support role, which can be claimed for depending on the tissue you are looking at or the cell populations you are looking at. A good potency assay needs to also perform as a good analytical tool, and that's why we needed to be quantitative as far as it can be, and to be robust enough for us to establish appropriate acceptance criteria for release. And since we are discussing release, that triggers another set of characteristics we need to obtain, which are accuracy, because we need that assay to support validation. But we also need, for those products that are usually fresh products, the method needs to be fast enough for the results to be available before administration to the patient. Obviously, sensitivity and specificity are characteristics you would expect for any good assay. And the fact that it needs to be predictive of the clinical efficacy is exactly what we've just discussed. That's the only assay that will give you insight of the potential of the product once administered. Finally, something that needs to be taken into consideration is even if the selection of those assays for a matrix may evolve over time, we need to have a set of parameters that will support comparability through the entire product development and ensure batch-to-batch consistency. The next figure is called Potency at the Core of Product Development, and this is something we had the opportunity to publish a few years ago already. What we see here is that potency assay, or the set of potency assays you may want to establish for release, are in fact derived from the characterization exercise, and over the development it's very likely that you will have back and forth with other potency assay candidates for which you will have characterization data only that may reveal to be more supportive for release, or that may reveal to be obsolete, especially considering what an assay should do at the time of marketing authorization. And something that is also captured here is that the entire exercise for most of the cell and gene therapy product is based on an assumed mechanism of action for which there is no complete understanding, even if we have sufficient level of characterization. The set of assays that will be retained for release will be then established as specification. And those specifications, as already mentioned, are key to establish product consistency and support comparability exercise. But potency is also one of your main stability indicating parameter, as an example. Finally, and that's a part that is not always easy, building the correlation with first the expected efficacy or the intended in vivo effect is really important for those assays. And whenever possible, they should fully support the dose. And we will see that depending on the type of product you are addressing, this may be more easy for some of them and a lot harder for other types of product. The next question that often comes up when dealing with potency assay development is, when do I need an established potency assay development? How do I address this in terms of timeline? And here we usually try to apply a tiered approach. As already mentioned, we need to have early potency measurement even at the stage of R&D proof of concept or early animal studies. No need for acceptance criteria, but we need to gather that initial set of data for long-term use and to start exploring that mechanism of action. That data captured early will also help us building a case of representativity of the non-clinical material versus the clinical trial materials should there be any change in the process at this stage of development. Finally, something that it's not often discussed but is critical is we need to communicate to potential investors that capturing potency early on is critical for the development and is in fact a way to prove that we are on the right track. Later in the development for the first in-human clinical trial, the goal here is to establish a first set of specification, and that's how you get approval for your initial clinical trial study from authorities. But at the same time, we need to anticipate additional potency assay candidates to complete the characterization exercise. So we will have that initial potency assay matrix which we just discussed with methods that may not be perfect in all aspects, and at the same time continue to measure additional potency assay candidates from which we will draw data later on to build that correlation between analytically performing surrogates versus poorly performing but highly representative of the mechanism of action qualitative assays. And that's where we will do between that first in human and the pivotal study that will lead to the marketing authorization or BLA. We need to refine that testing strategy by then, build that correlation between the surrogates, and support validation of the analytical method first, but also process validation. It's very likely that only your surrogates will be able to support that validation due to their characteristics. And that's why building that correlation as early as possible is really key, based on your manufacturing experience. Now is the time to try to share how we can address the challenges of those potency assay development and also the opportunities that we can draw from past development for each type of cell engine product. We start with the cell-based products here, as they are representative of a number of challenges with regards to potency. As an example, the biological activity isn't related to one signalization pathway, and we need to define very early what is our drug substance, what components are contributing to the mechanism of action, and have potency assay matrix that address as much as possible each of the contributive elements. It's very likely that the complete mechanism of action will remain unknown, but we need to build the potency assay around our understanding of that mechanism of action, which would have been explored by characterization studies and also the clinical results from clinical trials. Based on this, and taking into consideration the manufacturing experience to date, we will be able to establish a first set of specification that should be drawn from characterization. Finally, when dealing with the contributive or non-contributive cell population, it may be very difficult to discriminate which one are the one responsible for the activity. The way to address that challenge is usually to go through extensive characterization of the cell population and not stick with the minimal requirements for identity testing. This needs to be done targeting potency evaluation, but also starting building that correlation, because in some cases, identity testing might become one of your surrogate for potency on the long term. Finally, we also need to consider potential impurities, meaning all elements that are not either contributive or supporting the mechanism of action should be considered as impurity, and you will have cellular impurities in those products that require monitoring. The general approach we have seen being successful so far is to build a matrix combining the identity, the viability of course, proliferation potential of those cells if this is part of the mechanism of action, and also build the correlation towards a targeted secretion profile that is supposed to be the signalization pathway of your activity. The next product category we wanted to discuss are the tissue-engineered product. While most of the cell-based product challenges could apply to tissues, there are a number of specificities that make this category separated in terms of potency assay development. The first thing is that part of the mechanism of action for the tissues is already known compared to cell therapies, meaning those tissues are usually here as repair or replacement of damaged tissue, and their mechanism of action is essentially to be as close as possible to the tissue we want to replace. Also, part of their mechanism of action relies on structural properties, whether they are inherent from the tissue or whether they are brought by a combination with a medical device, matrix, or any type of supports that could be used in the manufacturing. The tissues also often a qualitative method that is used early in development and that should be used to build correlation, as we just discussed, which is histological analysis. the understanding of the tissue histology is usually well documented, and comparing the tissue-engineered product to actual representative tissue is considered as contributing to the potency assay test. In terms of challenges, some things that cell therapy may offer, especially when they are allogeneic cell therapies, that tissue usually cannot offer, is the fact that we are limited in the number of samples we may obtain from a tissue. Or, in other terms, for destructive testing, we cannot test the entire tissue, and we need to have approaches with a very specific sampling strategy, whether we take a part of the construct, whether we have coupons used for release. It is in this case very important to also build correlation between the testing sample and the rest of the product to support releasing a product based on partial testing. Finally, dose definition may be easier for tissues than for cell therapies just because we usually have a dose expressed in terms of piece of tissue or size of the tissue to be applied. As a general approach for tissues, we try to use markers that can be common to both histology staining and gene or protein expression from the cells to correlate what we observed in the qualitative histology with quantitative measurement. Last but not least, the final categories of ATMPs would be the gene therapies. And while they are often considered more straightforward in terms of how to build the potency assay, especially considering the fact that the process to obtain those gene modifications is closer to what we are used to see for recombinant biologicals, as an example, those products also present very specific challenges that are related to that potency assay strategy. The first element is there is a general understanding that compared to cell-based or tissue product, we may have more safety concerns when we deal with gene modification. A good example is that the vector toxicity should be addressed as part of the potency, whether it's toxicity for patients or for the cells we are trying to modify ex vivo. Similarly, the integration and potential for off-target integration of such gene modification is required as part of the potency assay. The overall vector design and the serotypes that we have selected if we want to claim it participates to the mechanism of action is also critical as part of the potency assay. And finally, a lot of those gene therapy products are in fact ex vivo gene therapy, and as such they contain cells, and most of the challenges we have seen for the cell-based medicinal product will apply to that category. The general approach we've observed over the last decade is that for gene therapy, usually potency assays are built around vector integrity and infectivity. And that's the basis for potency evaluation. And then as part of the complementary studies, we need to keep in mind that the potency will always be considered in the light of safety for those gene-modifying products. As a conclusion, as we've seen, it's not an easy thing to develop a suitable potency assay for this type of product, and we still need to do it because this is in fact the only critical quality attribute that is predictive of the clinical efficacy. The development strategy should be addressed as early as possible in product development, and it's not something we need to do to fulfill compliance with regards to authorities' expectation, but also consider this as critical for the overall quality assessment and critical for the characterization exercise. If it's anticipated that potency assay strategy will allow to build correlation for a sustainable strategy of release on the long term, using surrogates that will be sufficiently quantitative for validation, but also fast enough for commercial release, while ensuring that we will be able to release product in a cost-effective manner. Thanks a lot for your attention. If you have any questions, feel free to contact us. There is an email contact at the top of the poster. I wish you all a very pleasant day. Thanks again.